let's chat about metabolism. Well, first, let's define what it is. Metabolism. How would you describe in your own terms? Okay, that's it? Don't look there, it's boring.
Burning fat cells and other cells.
Okay, it's all breakdown.
Breakdown of everything for energy.
Okay, and that's it. What do we just release energy like steam engines? What do we use it for? Energy. Okay, movement, huh? Well, what do we do? Well, we definitely don't do it anymore. When we were younger, what we used to do? Yes, grow. So it's breakdown, right? For energy production, then use of that energy for synthesis, for growth. Does that make sense? So all chemical reactions, everything, all chemistry of the body is called metabolism. Okay, we can split it into subgroups. You know, scientists love to put things into boxes. So anabolism is when you synthesize things. Catabolism is when you break them down into the smaller pieces. Usually catabolism is when energy is released, and anabolism is when energy is used. Okay? That makes sense? There are actually studies now. There's a separate, I wouldn't say it's science, but area of biology that is called Metabolomics. You may sometimes, see that name. Metabolomics is the study of all chemical products that organism makes. We now have technical capacity to collect, say, a population of bacterial cells and analyze them and collect data of virtually all chemicals that they make at a given moment. It's enormous amount of information if you think about this. It's huge, but if we compare the chemicals that are present in the population or a cell, well, usually it's not one cell, it's a set of cells. In different conditions, we can say, okay, these conditions trigger the production of that chemical. These conditions trigger the production of that, and what we can derive, what conclusions can we make out of it. It's now the time of big data when people look at the massive amounts of information, and of course they don't just look, software does it for them. But you can now, with technical capacities that we have, we can analyze, compare enormous. We don't have to look for a specific molecule. We can look at all the molecules that we have. Now, you mentioned energy, right? So we have to use something as the energy source. What we use as the energy source? Mainly. Huh? Before, we have to make it. Yes. Well, mostly sugar. We can use fat. We can use food, right? So we use food as a source of energy. There's something else we have to get from the environment to become bigger. Or microbial cells have to get from the environment to grow. Energy and what else? Nutrients, right? And nutrients means carbon. Because everything, all organic matter is made of carbon. And when we say made out of carbon, look, everything is organic. This plastic box that computer's in is organic, absolutely, it's organic carbon. Bananas, 49 cents in Walmart, are organic. The apples for 15 bucks a pound from Whole Foods, no doubt organic. They organic in different way, not conventional organic, but it's still organic. It's all organic carbon. Okay, and on the other hand, we have inorganic carbon. Where? Where can we find inorganic carbon around us? Good. Okay, I'll take it. Ash. Well, in ash, actually, not anymore. Ash is mostly potassium hydroxide. But where do you take? Well, coal is kind of hard to advocate. But where else can you get inorganic carbon? Plenty of it in this room just now. Yeah, CO2, right? CO2 is the major source of inorganic carbon. Okay? That makes sense? So we figured out where can we take carbon, which is a major nutrient. Now we need to figure out where can we take energy. So energy can be taken from light. Does that make sense? Or it can be taken like we do from chemical sources. That makes sense? When we burn coal, we get energy from chemical sources. When we eat food, we produce energy from the chemical sources. So based on those categories, the source of energy and source of carbon, organisms can be divided in several big groups. First of all, energy source. Chemotrophs versus phototrophs. Phototrophs obtain energy from light. Make sense? Chemotrophs obtain it from chemical sources like we obtain it from glucose. There are other ways, and we'll discuss them. Then we talk about carbon source. So if carbon comes from organic materials, can be food, what other sources of organic carbon can you think of? Other sources of organic carbon, except for like bread. It's all dead matter. Processed, actually. Well, dead. First of all, dead matter, right? Can you eat like a dead squirrel? Yes. Can microbes eat a dead squirrel? Sure. What about a live squirrel? Can they? Well, not me. I can't catch it. Well, let's focus on microbes. Microbes. Can they survive using carbon from a live organism? Yeah, how do we call them in this case? The ones that... Well, it's not necessarily only bacteria. Fungi can do that. Helminths can do that. How do we call organisms that get their nutritional sources from? Huh? Not necessarily. Virus is not an it's kind of tricky. But how do we call an organism that obtains its nutrition at the expense of a living parasite? Yeah. It's parasites, right? They can use living host as the source of carbon. Saprobes like fungi grow in stumps. It's a dead matter. You can get it from dead matter. Does that make sense? There are plenty of... Let me tell you something. For fungi, there's a joke among mycologists. If there is a source of organic carbon, there's going to be a fungus that uses it. Which means you throw away the polyethylene sac, there's probably going to be a fungus that will break down that polyethylene and will extract carbon from it. Fungi are extremely inventive. Okay, so those organisms that use chemical reactions as the source of energy and receive their carbon from organic sources called chemoheterotrophs. That's the most bacteria, practically all fungi, all protozoa, all animals with no exceptions. Does that make sense? can the same guys that can they use chemical sources for energy that are inorganic? Absolutely. So there's a group of microbes that sometimes are called lithotrophs, contrary to organotrophs, that use reactions between inorganic chemicals to gain energy. So for example, such reaction would be oxidation of sulfide ion. You can oxidize sulfide, produce sulfate, and that reaction will definitely yield energy. Okay. Now, carbon sources for them would be also inorganic, because in the places where these microbes live, the organic matter is very sparse. Does anyone know where we can find the guys that can like get energy from hydrogen sulfide, from ammonia, some other crap? Yeah. Yeah, volcanic vents at the bottom of the ocean, other extreme environments. Organic matter is really not abundant there, so they have to obtain the carbon from inorganic sources. So they're going to be chemo, energy source, auto, carbon source, trophs. Does that make sense? Chemical source of energy, inorganic carbon. That makes sense? Now, phototrophs, they use light as the energy source. Okay? And if they get carbon from inorganic source like CO2, they're called photoautotrophs, and that's all photosynthetic organisms pretty much. Well, not all of them, but majority of them. Plants, algae, a lot of bacteria, okay, green and purple bacteria. Does that make sense? They pretty much they make their own food from inorganic sources. Okay. Photoheterotrophs are quite unique microbes that use light as the source of energy but obtain the carbon from organic sources. You can see that microbes span the entire range of opportunities for both the sources of energy and nutrients. Right? Now let's take a step back and take a look at, what energy source means. So when we are chemoheterotrophs, and I will have to use humans as one of the examples, because it's fairly easy. Well, okay, and some fungi, let's say yeast, are chemoheterotrophs. So yeast, what do they use as the energy source? Like brewer's yeast, what do they use as the energy source? Okay, when you make a moonshine, what do you have to start with? A mash, which contains a lot of what? Which nutrient? Huh? Sugar or other carbohydrates, right? Does that make sense? So it's carbohydrates so it's going to be CH2O. That's the general formula, like generic formula of carbohydrates. Okay. And what does yeast make out of carbohydrates? Ethanol. And in some cases, if it's a beer, carbon dioxide. So essentially, well, and they also will make some water. C2H5OH. Don't worry, it's not a chemistry class. I'm not going to ask you this. But yeah, if you're curious, go ahead. So what it essentially goes on there is that yeast, they oxidize carbon in the carbohydrates. Does that make sense? And that oxidation produces the energy for the growth. Okay? If you would look at the chemotrophs, you have, sorry, not plus, it's minus. Chemotrophs will oxidize sulfide ion, produce sulfate ion. So they oxidize sulfur and sulfide and transfer electrons from sulfide to oxygen. Okay? So it goes this way, electrons. Right? So if you would look at any process in any microbial cell that is dealing with energy, it's pretty much always about transferring electrons. Even photosynthesis. If you don't remember photosynthesis, I will remind you. The first thing in photosynthetic reactions, light hits the electrons and energizes them. And then those electrons go through electron transport chain. That makes sense? So we can picture it in this way. So you have some chemicals that you have in the beginning and chemicals you have in the end. Okay? These are electrons in those chemicals. So electrons move from high energy state to lower energy state, essentially producing energy. Does that make sense? It's all about transfer of electrons. Transfer of electrons from one molecule to another, or energizing electrons by the photons of light. Now, when we transfer electrons from one molecule to another, we call such a reaction how? Huh? Say again? Reduction or oxidation. Okay? Now, when something is oxidized, it means that this molecule loses electrons or gains electrons. Yes, something that is oxidized, like this guy, just lost electrons. Okay? And this one got reduced, okay, by gaining the electrons. So the one that pulls electrons away, the one that gains electrons, that takes them away from another molecule, is called oxidizing agent. So oxidizing agent is always reduced, and reducing agent is oxidized. Okay? Another name that sometimes are given to those chemicals, this is the reductant, the one that reduces, and this is the oxidant. Does that make sense? Now, what is the role of electrons in the molecule? What do they form, the electrons? The molecule that consists of several atoms. Electrons. The bond, right? So when you move electrons from one molecule to another, you inevitably break bonds and form bonds. Does that make sense? So if we will try to represent it in a pretty simple graph, like this one. If you go from this to this, the energy is going to be released. But if you want to form bond back, you will need to put energy in. Does that make sense? Now, electrons are extremely reactive. You've all heard about radiation, I believe. And there are three major types: alpha, beta, and gamma rays. Beta radiation is electrons. Okay, as simple as that. So you can imagine what chaos they can create in a cell. So you cannot let them loose in a cell. They cannot. So look at this from the standpoint of the cell as a space. Cell is pretty big on the atomic level. It's huge, actually. So if one molecule loses electrons in this corner of the cell, electrons can't just go by themselves to another corner. If electrons are lost by glucose in the cytoplasm, they cannot go to mitochondria by themselves. They have to be carried, like on a taxi. Does that make sense? So in the cell there are electron carriers, different types. One that you know almost for sure is NAD, nicotinamide adenine dinucleotide. Okay, you don't have to know the exact words for acronym, but the acronym NAD you have to know. So this NAD can accept two electrons. When it accepts electrons, is it reduced or is it oxidized? Reduced. So when NAD accepts electrons, it becomes reduced, and its reduced form for some reason is called NADH and always has hydrogen ion hanging out next to it. Why hydrogen ions? Wait, say it again.
Because hydrogen only has, hydrogen ion only has one proton, it can give up its electron to bond.
It not necessarily, I understand the idea, it's a great idea, but it's not necessarily hydrogen that gives electron away. What is the charge of electron? You can't just take two negative charges. You have to balance them always. You can't have unbalanced negative charge. So those hydrogens, they don't, when I say hangs out next to it, doesn't mean it's like walks along, okay? It means that in each reaction you have two electrons, two protons to balance out the charge. You can't just move electrons around. The charge should be zero. The sum charge should be zero. Does that make sense? Now, that's one. Question? No? Okay. That's one. Another one that you know about, probably FADH. Now, FADH can accept electron and proton, and that will give you FADH2. Its reduced form is FADH2. Okay? That makes sense? Now, all this energy flow, all this electron flow is essentially releases the energy, and this energy is then converted into the ATP. Does anyone remember, maybe even like very approximately, the oxidative respiration? Glycolysis and Krebs cycle and electron transport chain. Just, vague recollections, kind of vague recollections. Well, no. What I'm saying is like glycolysis and Krebs. They're like thousands of steps. And what in the end you have? You have ATP. Why the hell do we do that? Why we jump through all the hoops? Why cells jump through all the hoops to make ATP? Why? Why ATP? Huh? Why? Why do we need it? Oh, come on. Glucose can give you plenty of energy. You can burn pretty much when you oxidize glucose plus oxygen, it will give you water, CO2, and energy. You can burn sugar. You can burn glucose. Burn, I mean, with a flame. It will burn. Why ATP? Again? Oh, glucose is oxidized to CO2 and water. It's perfectly clean. Imagine that we got rid of all the money. Like money as a concept. So say Justin knows how to fix cars. And I know how to bake. I really know how to bake. I'm really good at it. I really need to replace my front struts. So I bring my car to Justin and tell him, dude, can you fix it? And he fixes it. And then I pay him. I don't have money, but I can bake. And I give him 50 loaves of bread, which is an awesome deal for me, not so good for Justin. I give him 50 loaves of bread. My struts are fixed. And he's sitting with 50 loaves of bread, which is a, let's imagine, fair price. But you're not going to eat 50 loaves of bread in like 2 days. Huh? So you either have to freeze them or use these loaves to pay for something else. So this exchange, can we direct exchange? Sure, we can. We carpool. I know how it works. I carpool on Tuesday, I carpool today, like other people carpool on Monday and Wednesday. So we exchange the service. Okay? But on the large scale, it cannot go this way. It's just not, it's not functional. Make sense? Now imagine, you have a cellular process, say transcription. It requires a lot of energy. And you get this energy not from ATP, but from glucose. So you got to have all the enzymes that will be very specifically sort of adapted to channel the energy from the glucose oxidation into the transcription. Translation is going to have a completely different set of enzymes. And God forbid you ran out of glucose. Now you have to think about how to extract that energy from lipids and then proteins. And for each cellular process that requires energy, you got to have its own set of enzymes specifically suited to this process. So it will become bulky and ineffective. Instead, you have a dedicated system, enzymatic system, that extracts energy from lipids, proteins, carbohydrates, produces ATP. It's one. Okay? And then you can use ATP in transcription, translation, replication, biosynthesis of arachidonic acid, whatever you want. Okay? Flagellar movement. Because in essence, ATP breakdown that's illustrated here is the universal process. There is one enzyme that facilitates it, ATPase. Does that make sense? So you use it as money in the cell to exchange energy between different cellular systems. Does that make sense? Now ATP, that's why we call it energy currency. It's not just a sophisticated, I don't know even how to say it, analogy. It's essentially currency. ATP carries energy in the form of chemical bonds between the gamma and beta residues of phosphate. So we call them gamma and beta phosphate bonds. Gamma bond carries more. So usually when ATP is hydrolyzed, when it is broken down, only one phosphate is released and you have ADP molecule. Okay? But you can technically break down one more and it still will produce energy. Does that make sense? Good. Now that release of energy during ATP breakdown, so energy is released, you see here? Energy is released. This is called exergonic reaction. Erg means energy, and X, remember, X means out, like exit. Okay, so exergonic reactions, they produce energy. They are usually reactions of catabolism, reactions of degradation. Does that make sense? You break down ATP, you release energy. Right? That makes sense to you? You break things down, you release energy. Now when you need to make ATP, you essentially put energy in the form of chemical bond. Chemical bond between the phosphate residue here and the rest of the molecule. So this reaction is endergonic. Okay? It accumulates energy of the sun or energy of other chemicals in the form of the chemical bond in the ATP that can be broken. Does that make sense? And if you would look at the reactions in the cell, we've already learned that reduction and oxidation are always coupled. You cannot have one without another. Turns out that in most cases you cannot have the endergonic reaction. You see the blue one here? The endergonic without exergonic. You gotta have one reaction that will release energy, like oxidative respiration. And this reaction will fuel up the energy accumulation, the synthesis. Okay, in this case, ATP production. Does that make sense? So the endergonic and exergonic reactions are coupled. Of course, nothing is 100% effective. We are not 100% effective. So all this energy that does not go into the endergonic reactions, where does it go? We'll lose it. We lose it. Microbes lose it in the form of it's the easiest way to lose energy. Lose it in the form of movement is the useful thing, right? Because microbes move somewhere. It's useful. I mean, lose it, just dissipate energy. Huh? Well, for microbes, breathing is not conventional breathing. Oxygen diffuses, and actually presence of oxygen allows them to do the metabolism, to perform the respiration. In what form? What form? We can't. Energy is not a material object. Heat. We lose it in the form of heat, right? Like your engine, your car engine. Technically, it's not supposed to be hot. It's a downside of an engine. It loses energy in the form of heat. We all lose every time. Every time we lose energy, we'll lose it in the form of heat. Does that make sense? So essentially, again, I want you to think about metabolism as the way to convert energy, to channel energy from chemical reaction to chemical reaction, or from light to chemical reaction. Okay, so it's all about energy transformation. I gave the Monday class example. Grass. When you seed the grass, what happens to it? It grows. Why it grows? If you will put it in a complete darkness, will it grow? No. So it accumulates energy of light in the form of carbohydrates and cellulose. Then you take a little calf and release the calf on the grass. And the calf starts to graze. And what happens to the calf? Calf. C-A-L-F. It grows into a big fat cow. Okay? Because it eats, consumes chemical energy that is in those starches and cellulose. That is a sunlight energy, okay, that was conserved there. Now we come and kill that cow and eat it. So we consume the energy in the chemical bonds in the protein and fat of that cow. Does that make sense? It's energy transformation, whether in one organism or in the bacterial, microbial communities in the entire earth. Okay. Now, energy, chemicals. Any questions so far? Chemicals, reactions. I'm going to give you a little question. I'm going to write down the simplest reaction that I can think of. 2 molecules of hydrogen, 1 molecule of oxygen will produce hydrogen and oxygen will produce Water. Exactly. Water. So, physical state of hydrogen is gas, oxygen. You can buy them. You can put them together in the glass tank. What do you think is going to happen next? What you going to observe? Nothing. I mean, unless you live like very, very, very long life, like hundreds of years. Reaction doesn't go. I mean, it goes very, 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 very slowly. So in 1,000 years, probably, all hydrogen and all oxygen will react with each other and form water. Okay? Another example. Nope. Yes. Okay. Match. Can it burn? What makes it burn? What else do I need for the match to burn? What another chemical, huh? Well, before friction. No. What? Like if I'm in a vacuum, oxygen. I need oxygen. So I have oxygen. I have match. It should burn, but it doesn't. So I need friction, right? Shit. Yeah. Do I need friction now? No. It burns by itself. Right? Why did I need friction in the beginning? Create a heat. Why did I need the heat? To initiate the reaction. If I would put this match to the mixture of hydrogen and oxygen, what would happen to me? Water mixture. I'm going to tell you something. I'm going to die. Because this mixture will explode, you know, and the entire oxygen and hydrogen mixture will produce a lot of water in a split second. But only when I put fire, temperature into it. So that was that energy that is required to start the reaction is called the activation energy. It's this green arrow that shows that bump that you need to go over in the beginning. Does that make sense? Once you pass that bump, the reaction pretty much goes by itself, like we saw. Okay? Right? This bump can be extremely high, like hydrogen and oxygen. Reaction between them, it can be very high. There are some reactions that you can observe that are much faster, like rusting. Like iron rusts, and it's a fast reaction. Oxidation, that is fast. Now, cell cannot afford slow reactions. Reactions should happen in a split second. So enzymes would that do that. Okay, enzymes facilitate the reactions. Enzymes reduce the activation energy. They make that bump smaller. That make sense? One of the analogies that I'm very proud of is Cinderella. I don't know about American version, but in the Russian version, there was something like stepmother gave her those stupid tusks, so she wouldn't go to the dance or whatever. And one of the tasks was something like stepmother mixed like a white rice and a brown rice, although it sounds more like Indian Cinderella, and told her to separate, okay, those grains. Now, imagine that you have that task in front of you. You have a mixture of brown and white rice, and you have to separate them. Can you do that? There you go. You can do it. I mean, it's just boring. You're not going to do that, but you technically can. It'll take you forever. And in that fairy tale, there are like little mice or some other creatures that come over and tell you, don't worry, we're going to separate it for you. Do it in like 15 minutes. Okay? Separate the rice. The same result. Okay? Grains are separated, but it's done much faster. In this case, mice, or whatever the creatures are, the enzymes. Okay? Enzymes facilitate the reaction. They speed it up. They increase the rate, but not the ratio. Which means we would have hypothetical enzyme that would facilitate this reaction. It would not change the amount of water that will be produced. It would only alter the rate of the reaction. Reaction would go faster. Does that make sense? Now going back to the Cinderella analogy. As far as I know, not a single mouse was dead after separating those grains. So enzymes are not wasted in the reaction. They are not chemically modified or anything. If you start with 10 molecules of enzyme in the beginning of the reaction, you're going to end up with 10 molecules of enzyme when everything is over. Does that make sense? Okay? Most of the enzymes, vast majority of them, are proteins, and for our purposes, we can think of all of them as proteins. There are enzymes, parts of the ribosome and parts of the splicing machinery that are RNA. But the enzymes that we're going to be dealing with are proteins. Does that make sense? Now, how do enzymes work? Basically, you have a substrate, the molecule before being converted, like a molecule of sucrose, okay, that binds to the active site of the enzyme. At the active site, whatever the structure is, this structure of the active site facilitates the chemical conversion, okay, of the substrate into the product. Does that make sense? So we can write it down as substrate is converted into a product, and this process is facilitated by enzymes. Now, enzymes are very specific. Sucrase will not break down lactose, and lactase will not break down sucrose. Although those sugars are pretty similar. Okay, how they are so specific? There are two models. They kind of are mutually complementary models. A lock and key model, that's the most popular. That substrate fits the active site, so lock fits the key fits the lock. But more popular now is the model of so-called induced feed. When substrate here interacts with the active site, it slightly changes its conformation. Does that make sense? So think about the key that kind of changes its shape a little bit as you put it into the lock. Does that make sense so far? Now, each enzyme works at the optimal conditions. For instance, enzymes of the stomach, like pepsin, what would be the optimal conditions? Huh? No, I mean, but chemically, pH. Acidic, not even slightly, like really acidic. Well, think about microbes. If you have a microbe that comfortably lives at the temperature of boiling water, enzymes will probably function better at the temperature of boiling water, but not at the room temperature. Does that make sense? Now, you have to appreciate that if we would try and graph the enzyme activity versus some sort of environmental condition, whether it's a temperature or pH, the graph will look like that, or like that, or maybe even like that, which means that there is a certain range of conditions in which enzyme is going to work, right? It's not just one specific temperature, but there is a peak, okay? That's the optimal temperature, optimal pH, or optimal salt concentration for the enzyme. Is that clear? Enzymes can be produced inside of the cell, and then they are called endo enzymes. Those will be enzymes that are dealing with the cellular issues, I don't know, replication, transcription, translation, biosynthesis, I don't know, phosphorylation, degradation of the faulty proteins, and blah, 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 inside of the cell. Does that make sense? And there are enzymes that are released to the outside, so-called exoenzymes. So you see, enzymes interact with the substrates and release products. For the microbial cell, what would be the function of the exoenzymes on a big scale? What would they do? What are they needed for? Exoenzymes. Why release enzymes in the environment? What do they do there? How? That's a good answer. That's really a good answer. They optimize the environment. What would be the first need of any cell? Which it gets from where? For instance, chemicals. Yes, chemical sources. Now, usually microbes don't live like in the sugar. Okay? If you think about fungus that grows on, say, an apple, when fungus grows on the apple, apple becomes kind of squishy. It's softish. So fungus releases enzymes that break down starches and whatever carbohydrates there are outside of the fungal cell, so that products of the breakdown, mainly monosaccharides, mainly glucose, can diffuse into the fungal cell. Does that make sense? So, but that's pretty romantic. There are other situations when exoenzymes can become a virulence factor. For instance, there's an enzyme called streptokinase, which is produced by surprising streptococci. Okay, streptokinase breaks down fibrin. Can anyone tell me fibrin is the component of what? One of the major components of. No? Huh? Say again? Some bodily fluid. Fibrin is the component of a clot. Okay? So when microbe breaks down the fibrin, it essentially breaks down the clot. So microbial spread is not restricted by coagulation. Does that make sense? Obviously, if there's a person and all fibrin is broken down in the blood, there's no clotting, this person is royally fucked up. It's called usually disseminated intravascular coagulation. Okay? There are multiple complications from it. Okay, that's one. Another very common example of the exoenzyme is the condition called scalded skin syndrome. Has anyone heard of it? It's the condition that affects infants, newborns. The bacterial Staphylococcus aureus infects the skin and produces the enzyme called protease. Protease breaks down proteins, right? Now, this protease breaks down proteins of the basement membrane that separates epidermis and dermis. When basement membrane is gone, epidermal layer of the skin literally falls off. This is called scalded skin syndrome. So it's an enzyme, exoenzyme, that works as the virulence factor. Does that make sense? Now we figured out exo and endo enzymes. The next one is regulation. Enzymes can be constitutive, produced at the standard level, which does not which is not influenced by the levels of the substrate. Does that make sense to you? No matter if you have little substrate, if you have a lot of substrate, levels of enzymes produced in the cell stay the same. Contrary to this, a lot of enzymes are regulated. When, for example, increased amount of substrate will lead to the increasing amount of the enzyme. So you see the difference between constitutively produced and regulated enzymes. So how can we regulate exactly the enzyme production and enzyme function? We're going to start with the idea of inhibition. Inhibition, yes.
For enzyme regulation, does it work backwards too? So if you have more enzymes and there's more substrate?
No. But you can imagine that if the amount of enzyme is high, then probably the amount of product is going to be high. Now the product can actually inhibit the enzymatic activity. Exactly. We're about to get in there. Does that make sense? We're going to talk how enzymes are regulated. So one is enzymes can be, first of all, regulation means inhibition or activation. So enzymes can be inhibited by two major ways. Inhibition can be competitive and non-competitive. In the competitive inhibition, molecule of inhibitor binds to the active site instead of the substrate. Does that make sense? For instance, there's a chair, empty chair, and if Hunter and I both want to sit on that chair, that's going to be competitive inhibition. We have a competition. Does that make sense? Non-competitive inhibition is when the inhibitor molecule binds to another location, not active site, so-called allosteric site, that changes the shape, the conformation of the enzyme, and substrate cannot bind to the active site. So using the same analogy, if Hunter wants to sit on that chair but finds me so repulsive that he can't even come close to it. Okay? Sense? Now, interesting consequence and difference between competitive and non-competitive inhibition, if you think about this, if you simply increase the amount of inhibitor, you can outcompete the substrate. But if the microbe will increase the amount of substrate, it can outcompete the inhibitor. There's a drug that I believe you have heard about. It's called sulfonamides, different sulfa drugs. They sometimes called sulfa drugs, sulfonamide, sulfamethoxazole, other drugs of that group. They are competitive inhibitors. They structurally are similar to a precursor for folic acid. So they bind to the active site, one of the enzymes in a pathway for folic acid synthesis. And they inhibit bacterial growth and so on and so forth. That makes sense? But if microbe can overproduce that precursor, it can overcome the competition. With non-competitive, it's not going to work. You kind of have an optimal concentration. And no matter how much substrate is added, it's not going to outcompete. Does that make sense? Now, expanding the idea of competitive and non-competitive. So allosteric inhibition is what we've just discussed, okay? When inhibitor binds not to the active site, but to allosteric site. Same can happen for the activation of the enzyme. Activator may bind to the allosteric site and make enzyme more effective. Does that make sense? Okay? Now, another concept is the concept of holoenzyme and apoenzyme. I call them Batman and Robin. So apoenzyme is Batman and holoenzyme is Robin. Okay? Apoenzyme, Batman, by itself cannot do anything. It has to bind the, sorry, no, my bad. That's apoenzyme and that's cofactor. I misspoke. I'm sorry. So apoenzyme cannot do anything. It has to bind a cofactor. So Batman is so pathetic that it cannot do anything without Robin. When they are together, apoenzyme and cofactor, they form holoenzyme. Whole, the whole thing. Okay? One of the simplest examples that come to my mind is DNA polymerase 3. Remember the one that adds the nucleotides during the replication? Its cofactor is magnesium ion. Without magnesium, it doesn't work. I mean, absolutely not. It just doesn't. Another example is RNA polymerase, which consists, apoenzyme of which consists of 4 subunits, 2 alpha, beta, and beta prime subunit. Without sigma subunit, which is essentially a cofactor, it cannot start the transcription. Does that make sense? To understand the concept of apo versus holoenzyme, please know it. Okay? Now, other cofactors that we can think of, vitamins. Vitamins are pretty much all vitamins are cofactors for different enzymes. When there are no vitamins, microbes don't grow, humans get sick. Okay. Very often, and I would say mostly, enzymes exist in the form of pathways, which means that the product of one enzymatic reaction serves as the substrate for another one. Does that make sense? Like something in the manufacturing process. When, people do at a plant, this department does this, and then the product of that department goes to another department. You know these pathways. You know examples of the pathways. Glycolysis is the example of linear pathway. Krebs cycle is the cycle. Okay? Heterolactic fermentation is the separation. Okay? Splitting pathway. Now, in many cases, the product of the entire pathway, here, end product, will serve as an inhibitor for the enzyme that is in the very beginning of the pathway. Does that make sense? Because when amount of product accumulates, this product will inhibit the initial part of the pathway to stop it altogether, okay? Working in a negative feedback fashion. That makes sense? The idea of inhibition, like this. Okay. Can take a break.